as mentioned, I do cancer research mainly focused on breast cancer, and I work in the interface of basic biology and clinical science of breast cancer. So it's called translational research. Are you able to see me and my screen, or only my screen?
Yes, ma'am, both.
You both. Okay, that's good. So this interface translational research I've been working on for like about 10 years now. So my training is in basic biology, but now I interact quite often with clinicians. So I get to see what is the real world problem in cancer and how can we add research angle to it to address those real world science clinical problems related to breast cancer. So my talk, the first talk, is going to come from therapy angle of breast cancer. The basics of it we'll talk about in the next lecture. The reason I chose this sequence is because from basic science background, we always think about how things develop and how things go wrong. Yeah, that's our direction of thinking, and that's how we are taught to think in basic sciences. In application world or in clinical science, the doctors mainly are taught this is the problem, think about how to solve the problem. Yeah, they don't worry about how the problem occurred, right? As a translational scientist, we try to bridge this gap in thinking also. Yeah, we not only want to understand how the problem occurs, we also want to find a solution or address that problem, find therapy for that problem after understanding what goes wrong, how things go wrong, right? So in this first lecture, I've tried to bridge both the angles, right? What things, what signaling pathways, what genes are going wrong in cancer biology, and what angle people use to address, to mitigate whatever is going on in cancer biology. Yeah. Next lecture, we'll talk about how things go wrong. Yeah. And why things go wrong. As of today, we do not have any therapy in any disease to prevent any disease from happening. Yeah, we understand in many circumstances how cancer arises, but we do not have any control on how we can stop cancer from happening. Although a lot of labs, a lot of institutes are invested in working towards, and we'll touch on some of the angles of those in the next lecture. Yeah, how can we prevent cancer? The first talk will be how do we target cancer based on what's going wrong in the biology of cancer. Yeah. So there are a few sections I have. This is based on the cancer hallmarks. They have been evolving. This paper that I'm referring to is the second one. There is a third one also that has come out 2021. That's way more complex than this one. So I am sticking to the second one. The first one is simple. It has only 6 different pathways that contribute to cancer formation. The one that is recently come out 2-3 years ago, that's far more complex. I feel this one is simpler enough to explain and still touch on the complex things that have been coming across, right? General theme though, I'm going to talk about few of these pathways into detail, right? So after each section, we can take a break, and if you have any questions on that particular section, you can hop on, right? Ask me, and then we can move on to the next section. We don't have to rush through every single thing. How do people ask questions here? Do they raise hand or do they start talking?
Both ways.
Yeah, so I am able to see only my presentation. I cannot see anybody's faces or hands. So it will be good if you just stop me and start talking and ask me a question. Yeah, if you have any burning question at that point. Yeah. Okay. So let's start with the hallmarks of cancers, right? So now this is the central dogma you can call about the way we have central dogma for life from DNA to RNA to protein. That central dogma actually translates into the central dogma of cancer. Any processes that go wrong in the central dogma, you have abnormal growth, and that abnormal growth can eventually lead to cancer formation, right? So what are the different factors that contribute to cancer formation, right? So one of the major pathway that has been discovered and long been known is sustained proliferative signaling, right? Any proliferation signaling is must for cancer cells to progress further. Now, there are multiple different pathways depending on which cancer subtype we are talking about that fuels sustained proliferation. Human cells have a limited lifespan, right? And it is determined based on the telomere length. So after certain number of replications, after certain number of cell divisions, cells stop multiplying. Either they cease to divide and maintain their function, or go senescence and die, right? This evasion of ceasing of proliferation is what is sustained proliferation, and that's what leads to cancer progression. So that's what we're going to talk about first, and we're going to look at various different signaling pathways that contribute to evade this senescence or halting of proliferation, leading to continuous proliferation, and what are different drug mechanisms that we employ as of today to inhibit this continuous proliferation process in cancers, right? So there are various different signaling pathways. These are primary fundamental backbone of proliferation. Now, depending on the cell type, depending on the organ system, there will be different factors, different receptor families, different kinase cascades that will be playing a role in proliferation, right? The major ones, though, we call them RTKs, right? So the tyrosine kinases. So major ones are epidermal growth factor receptors, ERBB1, which is EGFR, ERBB2, which is HER2, ERBB3, HER3, and ERBB4, right? Although these EGFR receptors belong to one family, each of these has a very specific organ specific expression. Yeah, for example, EGFR1 is mainly lung specific expression. It is expressed in many different organs, but the predominant function of EGFR1, which is ERBB1, is in lung. ERBB2, the gene, translates into protein HER2. And all of us know that HER2 amplification is responsible mainly in HER2 positive breast cancer. Yeah, so there are three types of breast cancers we have. The one that is driven by ER expression, estrogen receptor expression. One that is driven by HER2 amplification or overexpression of HER2 gene, right? So this HER2 is predominantly found in breast cancer, about 25%. There are other cancers where HER2 is also overexpressed or amplified. Those are gastric cancers, those are brain cancers. But in India, we rarely see any gastric cancer cases, mostly in the Asian countries like Japan, Korea. That's where gastric cancers are far more. And in those gastric cancers, about 10 to 12% have HER2 amplification. Brain cancers worldwide don't have that much incidence, but in breast cancers also about 2 to 5% of brain cancers, have HER2 amplification. Now, HER3 is a signaling partner that works with EGFR or HER2. So by itself, HER3 is never overexpressed in any of the organ system, but HER3 plays a major role in drug resistance. How it will, we'll talk about it later, right? And HER4 is also a partner mostly in the brain organ system. So when we target any brain related cancer systems, HER4 is what comes out in play. Now, fibroblast growth factor receptor, FGFR, FGFR2, come into play when there are mostly muscle related cancers that are coming into picture. Yeah. Then we have platelet derived growth factors, CSF1R, SLFR, c-KIT, and CFMS. These are the targets mostly in blood related cancers, right? Insulin growth factor, again, it comes on and off in various different cancers. Neurotrophins, mostly in brain related cancers. So as you can see, the drivers of hyperproliferation, that is increased proliferation, can be anyone, and there are many more which I have not listed. Yeah, this list is not exclusive. There are many more that play into role. But the general theme here that these tyrosine kinase receptors have a similar fundamental structure. They have a cytoplasmic domain, they have a transmembrane domain, and they have a kinase motif inside the cell, right? So most, because these are membrane associated proteins and major drivers of proliferation, these have been easy to access, to target with antibodies, yeah, or small molecules, because this organ system, cellular system is so compact, the drug delivery and access to drug has been always a major problem in cancer systems. Like, for example, the cancers that are buried deep inside, like pancreatic cancer, brain cancer, where there is a blood-brain barrier for the drug to be accessed, it is very hard to target such cancers, right? That has been the case long along, and people have circumvented those accessibility problems by targeting proteins that are outside the cell, that are accessible to any molecules that we intervene into human system. So most of the antibodies that are developed, or small molecules that are developed, those target the outer domain, the domain of these receptor proteins, right? So various different mechanisms that these hyperproliferative pathways employ to increase proliferation, and this is a kind of boiled down summary of various different means and modes that they employ. So the basic fundamental one is increased expression, right? Just make more number of copies of these transmembrane and kinase domain proteins, and because you have hyper activity of kinase signaling, the downstream effect of these kinase cascade is increased, and you have hyperproliferation into the system, right? Or you have mutations, point mutations, which remove the inhibition of these proteins, leading to hyperproliferation. Yeah, same hyperproliferation is mutations are achieved by point mutations or insertions. Overexpression is achieved by either overexpression of mRNA or direct amplification at DNA level, the way we see in HER2 cases, right? HER2 gene gets amplified. So the rare mechanisms, nevertheless, they exist, more so in blood related cancers, not so much in solid tumors, is translocation or combination of two different proteins. So for example, we know one protein in blood related cancers is RUNX1 ETL, right? So that example I have not listed in here, but RUNX1 gene is a transcription factor. It has a TAD domain, transcription activation domain, which binds DNA and leads to whatever gene activity. It has C-terminal domain, which binds to some partner which inhibits RUNX1 activity. So what that gene, what the beautiful system that has done, that the C-terminal domain is deleted, and that RUNX1 gets fused with another gene which has active kinase domain, right? And together they achieve hyperproliferation in the system because both of the inhibitory domains of both of the proteins are removed. Same way, just removing the inhibitory domain of EGFR or NF-kappa B is sufficient to achieve hyperproliferation. So these are various different mechanisms. The kinase cascade is amplified by overexpression or tiny alterations in the key kinase gene, key kinase protein that's responsible for these hyperproliferative pathways, right? So this is one simple example of how the very first targets that were developed to inhibit this hyperproliferation, right? So this is an example of EGFR, which is HER1, ERBB1 gene, is always has hyperactivity when it homodimerizes. EGFR can dimerize with HER3, HER4, very rarely with HER2. But the homodimer is the one that is most active, right? And the very first antibody that was developed to target this EGFR, anti-EGFR monoclonal antibody, would bind only when EGFR is homodimerized, right? So when the antibody is bound to homodimer of EGFR, it will inhibit the ligand binding. And when ligand cannot bind these homodimer receptors, the downstream hyperproliferative signaling, which goes through PI3K, AKT signaling, and KRAS and ERK signaling, will be inhibited. So this is the most simplified view and the very first few drugs that were developed to bind the receptors outside the cell membrane, which will inhibit ligand binding and inhibit the downstream signaling of these kinases, right? So there are, as system evolved, there are few different inhibitors that have come about, not just monoclonal antibodies. So like cetuximab and gefitinib. So setrusumab is an antibody, right? So gefitinib, erlotinib, lapatinib, these are the small molecules that were developed to inhibit ligand binding to EGFR receptors. Now, there's a simple rule to understand which one is an antibody, which one is a small molecule. When you have AB at the end, it's an antibody, and when you have IB at the end, it's a small molecule inhibitor. Yeah. So, as the system became more advanced, as we started to understand how signaling works better, and as more and more small molecules that were developed, which can pass through the cell membrane, the targeted therapy to target the kinase domain of these EGFR receptors that came about. As you can see, all the inhibitors that target kinase domains are small molecule inhibitors, right? Dasatinib, midosaturin, and whatnot, right? So either you inhibit the ligand binding with extracellular membrane inhibition, or you stop the phosphorylation or dephosphorylation of the kinase domains of EGFR directly, or the further downstream kinases in the pathway with various different inhibitors. Now, we will go and see further, uh, as we try chasing the cancer and inhibit whatever signaling that we have understood has gone wrong. Cancer does not stop there, right? It also chases further and tries to evolve to evade whatever inhibitions that we have been developing. And this evolution of cancer after you treat with a drug and it still recurs is what has driven development of so many various therapies, or so many various drugs to target one single molecule like EGFR or downstream signaling of EGFR, right? So these different inhibitors are not used as single agent at a time, depending on the person, depending on the type of cancer, depending on whether we know what kind of mutation this person has or not. A combination of therapy is also used these days. Yeah. This is just a small glimpse into how complex things will evolve as we go further. So far, so good. We have any questions? Okay, so we'll go further. Now, this is the whole trajectory of where we are today. This is only with respect to EGFR inhibition. Yeah. EGFR targeted therapy is mostly employed for lung cancers and also colorectal cancers, where EGFR is shown to be either mutated or overexpressed. And the very first monoclonal antibody, the cetuximab that we saw, was developed in 2003. And as we come along until this is still 10 years ago, we are still 10 years far away, further apart, there are so many different second line, third line therapies that have come across to overcome the resistance that cancer showed to the very first monoclonal antibody that was administered to cancer patient, right? So this evolution of targeted therapy continues even as of today, and how is that what we are going to see further, right? So you target the extracellular membrane, the ligand does not bind EGFR, and the kinase cascade is stopped. Hi. What is this?
Some students raise hand. I think they have some questions.
Okay. Yeah. Can go ahead, please.
So many back is so many. You have some questions.
You can ask me a question. If not, you can keep it written down, and we can come back to it at the end of the seminar also.
Okay.
Yeah, because I'm not able to see anybody right now. Okay. So the challenges, as I said, are still there. So once the kinase cascade stops, the binding of the cetuximab antibody to EGFR receptor is not permanent, right? It's a reversible binding. So cancer has a way to evolve out of these inhibition by various different ways. Either the same EGFR receptor shows a mutation in exon 20, or the parallel pathway through KRAS activation gets activated, which leads to hyperproliferation because of inhibition of EGFR, or the PICK pathway that goes through PTEN, so is blocked because of PTEN downregulation, and EGFR inhibition stops working, right? Or there are many other ways to circumvent this inhibition that we apply through EGFR inhibition, is the downstream activities of these kinase pathways go further. Excuse me?
Bhagyashree Somani, please mute your audio.
स्लाइडवर कडेला घ्या.
Bhagyashree Somani, ma'am.
Okay, unmuted.
Okay. So there are ways that the cancer will evolve further, and the treatment that we have given is going to be rendered useless after a few administrations. So um, uh, it is important for us to not only develop therapies but also to understand how these therapies are targeting the current pathways, and how these signaling pathways are intricately linked with various different signaling happening inside a particular cell. We are still in the middle of understanding how the signaling crosstalk happens, and actually the drug resistance that develops in cancer system is a very good window for us to understand which different pathways talk to each other and which different pathways can compensate if we inhibit one function, right? So such combination, like targeting these various different pathways that cross talk with each other, has been thought about already. And one such combination that can be targeted is, along with these signaling, we also target DNA damage response, as you will see further. So we in a given patient today, because of NGS DNA sequencing, we are very well aware of the repertoire of mutations that are present in that particular tumor, in that particular person. Germline mutations that are inherited through families, or the somatic mutation that tumor acquires as it grows further, or the tumor acquires as it develops resistance to given therapy, right? So we know the genetic mutations. We also can do, as of today, transcription profiling and look at what kinds of genes that are expressed in that particular tumor that are leading to either hyperproliferation or inhibition to the therapy. But what we do not know is how these different mutations and the pathways that are altered talk to each other, right? So and because of this lack of understanding, we still stumble upon the resistance that comes across with various single agent therapies that stop working after a while, right? Because the cancer cells are smart enough to rewire their signaling pathways to circumvent whatever inhibition we are approaching. I gave you one example of anti EGFR therapy, how the various different resistance mechanisms that come across. But that's true for every single therapy that's available today. Yeah, anti-HER2 therapy has various different resistance mechanisms. Even anti-estrogen therapies have various different resistance mechanisms. And today, in most of the cancers, the challenge is not the therapy not responding. The challenge is recurrence of the cancer because of resistance to the therapy. So, as of today, I would give you an example in breast cancer. 80% of the cancers, breast cancers, are treatable. Yeah, 20% of the cancers are a problem because either they defy the therapy, they do not respond to the therapy, or recur back because of these secondary mutations and secondary signaling problems that come across. And major inventions and discoveries and pharmaceutical companies are invested in to circumvent the resistance to the therapies, uh, to whatever that we have available as of today. Yeah. So this is like a second line therapy that came across to target the second mutation that occurs after the first line therapy. So we give cetuximab, which is EGFR antibody. Down the lane, the cancer develops mutations in EGFR gene itself, and the therapy stops working. And we give a second line therapy which targets that mutation that has arisen in that particular person. For example, osimertinib, which targets the exon 9 deletion and exon 21 mutations in EGFR. Right. Now these new drugs that we are smarter than the previous one, which bind irreversibly to mutant EGFR. So because it is difficult, the reason the first line reversible EGFR tyrosine kinases were developed because EGFR is expressed in many different organ systems. So if you inhibit irreversibly EGFR signaling, your the colon system is going to stop working, your gastric system is stop going to work, your hair system is going to fall out, because all these stem cells need EGFR for normal functioning. Yeah, and that was the reason the first line therapy that was developed, that was reversible. So all these non-wanted effects can be tailored down. The second line therapy, the irreversible EGFR inhibitors that are developed, those bind to the mutant EGFR, the mutated EGFR. Now, mutated EGFR, the EGFR mutation happens only in the cancer tissue because cancer is evolving because of the targeted therapy to EGFR, and these mutations are acquired only in the growing tumor, not in the normal body organs. Right? So this second line therapy, which targets mutations in EGFR, these are irreversible binding inhibitors. Right? So they stop the second resistance that is acquired within the tumor by irreversibly binding to these mutations, thereby completely inhibiting even the recurrent tumor. Right? So there is a third version of irreversible inhibitors, and the fourth version that has come across is, along with the irreversible inhibitors, also tag along whatever secondary side effects of proliferating tumor happened. So for example, here they gave anti-VEGFR. VEGF is a growth factor receptor and VEGFR is a receptor for that. Both of them in combination lead to angiogenesis. So endothelial cells express VEGF receptor 2, and expression of VEGF will induce more angiogenesis in growing tumor. Now, all of us know if more blood vessels are developed in a growing tumor, it can avail more oxygen and the proliferation can go further up. Right? So the fourth line therapy that was developed was a combination, not only target mutation of EGFR with irreversible inhibitors, but also inhibit the new angiogenesis that's happening inside the tumor. Right? So along with EGFR, target VEGFR also. So this is where the combination therapy started further on. Now, so in one of the slides, I talked about not only the mutations within the gene that we are targeting, but parallel pathways that can evolve to circumvent the inhibition that we are applying. So another mechanism, EGFR proliferation inhibition can be circumvented, is activating mutations in KRAS, which is a transcription factor that can take proliferation further, right? So KRAS has been about 20-40% of lung cancers have KRAS mutation, and these mutations occur mostly in resistant the lung cancers that are resistant to therapy, right? And KRAS has been elusive for drug development for about 20 years. There were like almost 18 companies that were invested in developing a drug against KRAS because this was one of the major resistant mechanisms that was in lung cancer. And in 20 years' time, they are able to develop the first drug, and this was celebrated quite widely in the cancer community, that they are able to target one single mutation in KRAS, that is G12C. Yeah, which is quite a potent mutation Actually, in our basic science lab, we use this mutation to transform the cells. You take simple mammary epithelial cell line, express KRAS G12C gene, the KRAS with G12C mutation, and the cells will transform into cancerous. Yeah, that's how potent this one single mutation in KRAS gene is. And in 20 years' time, they were able to develop a drug, a small molecule inhibitor, sotorasib, that binds this specific single mutation of KRAS. Yeah, and so this was in 2020. People were working on this for 20 years, 2000, like since 2000 the companies were invested. And as you can see, most of the efforts in therapy development for cancer um, are based on which particular cancer is predominant in that particular region at that time, right? So as we started understanding that cancer happens, we can target with therapy. When we stop the proliferation, cancer stops growing. When this understanding came about, people started documenting in terms of what is the cancer incident, what not. This is, I'm going a little bit side from the biology aspect, but this is important to understand in the clinical context. Is lung cancer was one of the most prevalent cancer for long period of time in the world, including India. Yeah. um, as of today, situation is different, but until 3 to dominant cancer. That is still the case in the West. As of today, situation is different, but until 3 to 4 years ago, lung cancer was the most predominant cancer. That is still the case in the West. In India, though, since last 2 years, the first most predominant cancer has become breast cancer. Yeah. So as of today, we have the highest incident of cancer in breast cancer. At the same time, lung cancer, there are so many therapies that have been evolved because it was predominant, and most of the pharmaceutical efforts that were put in mitigating lung cancer, we have very good line of first line, second line, third line, fourth line therapies to address lung cancer situation. So almost 80 to 90% of lung cancers are treatable, curable, even after they recur back. The mortality because of lung cancer as of today is reduced quite a lot because of these efforts that have been put in to address the most predominant cancer in the world. Right now, today the efforts have been directed towards the cancers that have coming up as more prevalent, like breast cancers. There are cancers that are still elusive to therapies, like pancreatic cancer. Pancreatic cancer being very buried inside the body, number one, it's very difficult to diagnose, irrespective of which part of the world you are in. And because it's buried deep inside, it is also difficult to target with drugs. Right. We still don't have good handle on pancreatic cancers, and there are very few labs invested because it's a problem that it is difficult to study cancer. Right. Breast cancer is high prevalent, accessible, and now last few years the efforts that have been directed are predominantly trying to mitigating breast cancer. Yeah. Sorry for this sideline, and we'll continue with our next line. So, after sustained proliferative signaling, as you undergo hyperproliferation, you're trying to go through your DNA replication faster and faster, and that itself is a cause to bring in more genome instability and introducing more mutations in the system, which leads to even further secondary tertiary mutations that are gained. And because of the survival of the fittest, the mutations that help you grow further get fixed, and the tumors with clones of various different mutations are formed with various different abilities, evading abilities to drugs and to continue growing further. Right. So how do we address this genomic instability that leads to more and more secondary tertiary mutations in the growing cancer, or the cancer that is recurrent to given therapy? right? That's what we're going to talk about. Now, the basic fundamental principle that we use here, that anytime there is a DNA damage for whatever reason, due to hyperproliferation, because a replication is happening fast, or a single line agent like UV and whatnot, right? These mutations are recognized by PARP proteins, poly-ADP ribose polymerases, and these PARP proteins will recruit various different pathways to remove the break and fix the DNA, right? If these DNA replication, DNA damage response pathways are functional, the mutation or the DNA damage is fixed, and cells undergo normal proliferation. But if this is not fixable, then the cell undergoes apoptosis, right? Now, we are inhibiting this PARP protein, which recognizes this DNA damage, right? So, if PARP is not recruited to the DNA damage site because we are inhibiting it, the cell will look at it as a signal to undergo apoptosis, because PARP is not recruited, none of the DNA damage rescuing pathways are recruited to rescue this DNA damage, right? So when we inhibit path, it's a straight away signal that the cell has to undergo apoptosis, and the cancer cells eventually die. So this is the basic principle employed. So you have a first line agent which inhibits hyperproliferation, for example EGFR, target EGFR. The cancers will cease to grow, but the cancer cells that evade this inhibition and employ some other pathway to increase proliferation, these hyperproliferating cells are going to introduce DNA damage breaks, right? So if you inhibit recruitment of PARP, these hyperproliferating cells will be inhibited, will undergo apoptosis, right? So you have two targeting mechanisms simultaneously: inhibit the fundamental mechanism why cells are proliferating, and inhibit the checkpoint mechanism to continue hyperproliferation in these systems, right? So one of the examples of these checkpoint inhibitors is BRCA mutation, yeah, which impairs the DNA damage response. And once the DNA damage response is impaired, hyperproliferation continues. Yeah, so we want to stop this inhibition of inhibition, right? So most of the breast cancers have do come out because of BRCA mutation, and this PARP inhibitor therapy is employed where we know that germline mutations are the one that are responsible for DNA damage response. Yeah, that is the very first place these combination therapies that were employed. Patients that have metastatic breast cancer which has not responded to the therapy, and along with that they have mutations that remove the DNA damage response. Now, in these patients, if you inhibit PARP recruitment, then the cell senescence or the apoptosis is induced, and these patients responded very well to the PARP inhibitor therapy. And this was one of the very first trial that was run across countries, across continents simultaneously, and it was a huge success. That the combination therapy of PARP inhibitor in metastatic breast cancer, because as you can understand, metastatic breast cancer is, until this trial, is considered uncurable. Once you have metastatic cancer, breast cancer, you're done. There is no treatment available. Yeah. So this particular success of the trial was celebrated quite well, that even now today you can even treat metastatic breast cancers provided you have BRCA mutation or DNA damage mutation in the system, right? So, with that understanding that it's feasible to target two different proliferation pathways, people came back to the bench and tried various different combinations of therapies that target various different signalings based on what cancer you're working on, and various different drugs that induce DNA damage. Yeah, then you induce DNA damage, give PARP inhibitors, and that way kill the cancer, right? So this is one of the papers that talks about the combination therapy actually works well. Now this is on the bench. There's still time for it to go to the clinic and the trials and stuff, but the combination therapy together will induce the cell death up to 55%, right? So in principle, in cell lines, it's possible. Yeah. For the next step, we're going to talk about the epigenetic modifications that are responsible for hyperproliferation. Now, it is quite easy to understand that all these different pathways like hyperproliferation and DNA damage response or angiogenesis that happens through VEGF signaling, all these signaling are happening through protein expression, that happens to transcription, that happens to through whatever sequences, regulatory sequences that we have on DNA, right? So epigenetic modifiers are bound to affect every single aspect of these different mechanisms that lead to cancer progression. Now, there are many different types of epigenetic modifiers that either have modifications that affect transcription. The modifications are either on the nucleosome, like chromatins that are seen here, or we'll see further the modifications that directly affect the DNA sequence as well, right? So the DNMT proteins that actually lead to closing of the chromatin, right? So euchromatin, when you have hypermethylation, will go into heterochromatin system, right? When it's a heterochromatin, the gene expression is inhibited. And so whatever tumor suppressor genes mainly get inhibited, or whatever genes that are responsible to halt the cell proliferation further get inhibited through hypermethylation of those regulatory regions. Now when you inhibit these epigenetic modifiers like DNMT, this hypermethylation is inhibited, and the proteins that are actually good for cell death, like apoptosis and tumor suppression, continue to express, and thereby mitigating whatever hyperproliferating signals that are playing into the system, right? So there are various different inhibitors. So this whole line of thought, the epigenetic modifiers that came into picture, is because of the tumor suppressors, because people have been putting in effort to develop therapies to target that drives the tumor, right? And so far the checkpoints in the cell, the intrinsic checkpoints in the cell system, those are tumor suppressors, right? And as we looked at the resistance mechanisms can be intrinsic mutation in response to therapy, parallel pathways that get hyperactivated in response to therapy. At the same time, the intrinsic mechanisms are also deactivation of tumor suppressors. For example, P53 is one of the biggest example, right? 80% of all the cancers as of today have P53 mutation. Yeah, majority of the cancers cannot go further into hyperproliferative state without P53, without getting rid of P53, right? And there are various different modes of P53. Not only P53 mutation, you can also inhibit P53 expression. So, as when this was understood, the efforts were made in targeting these epigenetic modifiers. So, these tumor suppressors are not inhibited and they continue expression, helping the therapy of these cancers, right? So, not just DNMT, but there are HDAC inhibitors, various different inhibitors, HAT inhibitors that have been coming into play, which are almost always given as a combination therapy, not as a first line therapy, right? So one of the examples of combination therapy of these HDAC inhibitors, epigenetic modifiers, is in current immune therapy, which is still in clinical trials with anti-PD-1. We'll talk about this immune therapy further in detail. Yeah. How much time do we have? We have 10 more minutes. You have to wait for 10 minutes. Yeah. So, yeah, sorry. Now that's where we are coming to the immune activating antibodies that are developed to uh, to employ the immune system that is already functional in a cancer to target hyperproliferative cancer, right? So the very first immune therapy that not many people are aware of was developed to anti-CTLA4, and the current line of therapy, anti-PD1, PDL1. I'm sure you guys have heard about it. The very much celebrated immune therapy, which is targeting PD1, PDL1. How and what's the function and how that's what we are going to look at it now. So, before we go to this immune therapy part, I want to check with all of you if you have any questions. And we have only 10 minutes left. So, should we do the questions before I go to the next one?
Yes.